Hi class, this is our first series of lectures, the Introduction to Biological Psychology. I am your instructor, Dr. Nathan Morosky. Welcome. Our first lecture is called the Introduction to Biological Psychology, and our objectives for this lecture are to define what biological psychology is and identify some associated synonyms, words that you may hear describing biological psychology. I'll also describe the scope of the field and situate it relative to other branches of the brain sciences. I'm then going to name and briefly describe some of the most influential concepts, emerging topics, and future goals for brain research. But to start us off, I'd like to give a little historical introduction to show you some of the things that we're going to be describing, how people have examined the relationship between brain and behavior. So as a first case study, let's go back to the year 1861. The professor of anatomy at the University of Paris was Pierre-Paul Broca, pictured here. He was informed about a patient who presented with a progressive articulatory or expressive aphasia, that is, a disorder of speech production. Although the patient showed normal speech comprehension and had otherwise intact mental faculties, he could only utter a single word, tan, and that word tan became his nickname. Broca was quite interested in the case, theorizing that because of the progressive nature of the aphasia, a possible lesion, perhaps due to a stroke, might provide evidence for the cerebral localization of speech production, damage of area of the brain, and it disrupts speech production. Therefore, you can identify where in the brain produces speech production. Tan soon died, and Professor Broca was quick to obtain his brain, as neuroanatomists then and now are quick to do. Upon examination of Tan's brain, Broca identified a lesion localized in the left frontal lobe on the third convolution, area called the pars apicularis, anterior to the lateral sulcus, which divides the temporal from the frontal lobes. Broca would identify 12 additional cases of expressive aphasia, each of which presented with a lesion in this area, about 1/4 of an inch above your left temple. Broca had identified the cerebral localization of speech production. In the previous century, electricity was in the air, literally. Natural scientists, including our own Benjamin Franklin, was busy studying the properties of electricity. One physician at the University of Bologna, Luigi Galvani, pictured here, made a foundational discovery linking electricity and the workings of the nervous system. The story goes that one day Galvani was busy rubbing the skins of dead frogs together in an effort to produce static electricity to study its properties. A scalpel at hand took on a charge, and when Galvani touched the sciatic nerve of the frog with the scalpel, the frog's leg kicked. Each time he repeated the experiment, he could elicit a muscle contraction in the dead frog by applying an electrical charge to the nerve innervating that muscle. Galvani had discovered that nerves conduct information via electricity. Near the end of the 19th century, the great neuroanatomist Ramonique Hall demonstrated that nerve cells, those primary cells of the nervous system, are separated by tiny spaces, which we call synapses. If nerve cells do not come into direct contact with each other, then how is the electrical signal from one cell communicated to another? The answer came to a German pharmacologist in an unusual and now rightly famous way. It came to him in a dream. Otto Lowy, pictured here, relates the story. The night before Easter Sunday of that year, I awoke, turned on the light, and jotted down a few notes on a tiny slip of thin paper. Then I fell asleep again. It occurred to me at 6 o'clock in the morning that during the night I had written down something most important, but I was unable to decipher the scrawl. The next night, at 3 o'clock, the idea returned. It was the design of an experiment to determine whether or not the hypothesis of chemical transmission that I had uttered 17 years ago was correct. I got up immediately, went to the laboratory, and performed a simple experiment on a frog heart according to the nocturnal design. So what was this experiment that proved that neurons communicate via chemicals between their synapses? Lowy prepared 2 frog hearts in separate baths. He then stimulated the vagus nerve of one heart, causing its heart rate, or its beat, to slow down. He then took some of the medium, the stuff that the heart was beating in, that was slowing the heartbeat, applied that liquid to the other heart, And lo and behold, that second heart began to beat slowly as well. Lowy had discovered that neurons use chemical signals to communicate with one another. And in our last case is the famous case of Henry Moyllison. When he was 27 years old, he underwent a surgical procedure by the famed neurosurgeon William Scoville to treat the severe seizures Moyllison had suffered from since an accident when he was only 9. Scoville surgically removed the area of the brain and the temporal lobe that were the locus of the seizures. Moyllison recovered from the surgery, and yes, his seizures stopped. Unbeknownst to Scoville and other physicians, however, removal of the hippocampus and adjacent structures left Moyllison with a profound amnesia. For example, he would quickly forget anything, any person, including people he had just met. Moyllison is better known in the literature as HM. HM became the most studied patient in all neuroscience, and studies of HM's amnesia by Scoville and Brenda Milner have profoundly changed what we know about memory. First, that areas like the hippocampus are fundamental in memory formation. Second, that there are different forms of memory and that different brain structures are involved. HM's amnesia profoundly affected his ability to remember explicit memory, the type of memory where we explicitly state an event or episode. His implicit memory, that type of memory for skill acquisition, for example, was largely left intact. Moyllison's sad case reveals how brain and behavior are intimately linked together. These 4 cases illustrate the foundations of biological psychology, the study of the relationship between brain and behavior. Biological psychology has had a number of name changes over the years. For example, you may have heard it called psychobiology, and it's now more often referred to as behavioral neuroscience. All these names are synonyms with one another. Regardless of the name, the main goal of this field is to understand the biology underlying behavior and experience. Biological psychology is such a fundamental discipline because it seeks to understand and explain that which makes us who we are, everything from the study of basic reflexes to higher levels of consciousness. Because of the scope of biological psychology, the discipline is influenced, informed, and enhanced by multiple other disciplines, ranging from biology and chemistry to cognition and computer science. You can see in this description here that all areas of science tend to converge on the purview of biological psychology. Let's take a closer look at the brain itself, because we're going to be studying the brain in detail for this class. So what do we know about the brain? The adult human brain weighs about 3 pounds. If you were to put both of your fists together, looking at your thumbs, that's roughly the size of your own brain. It consists of about 86 billion neurons, or those are the primary nerve cells within your brain. And each of those neurons make connections with multiple other neurons and associated cells, forming synapses with about trillions of connections or synapses between neurons. It's responsible for every behavior, every thought, every memory that we have. As the brain controls behavior, behavior in turn alters the brain. The brain is the most complex system in the known universe. Given the complexity of brain and behavior, how do we even begin to explore the relationship between the two? Biological psychologists use several different perspectives in an effort to understand the biological basis of behavior. The 5 major perspectives include 1. Describing behavior, 2. Studying the evolution of behavior, 3. Observing the development of behavior and its biological characteristics across the lifespan, 4. Studying the biological mechanisms of behavior, and finally, studying applications of biological psychology. Let's see how these perspectives might be used to study memory processes. First, from a descriptive perspective, researchers might examine which areas of the brain are active during fear learning, how quickly fear memories develop, and how long they persist, like the development of a phobia, for example, or post-traumatic stress disorder. From an evolutionary perspective, one could examine how different species compare in terms of fear memory acquisition and persistence. Developmentally, researchers could ask at what age phobias first emerge. Mechanistically, we can ask What anatomical and chemical changes in the brain support such memories? And finally, researchers can apply what they've learned about fear memories and develop therapies to treat individuals with anxiety disorders and PTSD. This course is an introduction to biological psychology. We'll be examining and answering a number of questions related to brain and behavior. So as a preview of what's to come, consider the following questions. How does the brain grow, maintain, and repair itself over the lifespan? How does the nervous system capture, process, and represent information about the environment? For example, sometimes brain damages causes a person to lose the ability to identify other people's faces. What does that tell us about how the brain recognizes faces? What brain sites and activities underlie feelings and emotional expression? How does the brain manage to change during learning, and how are memories retrieved? The brain is wonderfully complicated, and although we are learning more and more each day about the brain, thousands of peer-reviewed articles published on the topic each year, there are a lot that we don't know, a lot. But that's a good thing. Our current ignorance about brain and behavior relationships motivates all behavioral neuroscientists to ask and answer questions. Perhaps due to its complexity, perhaps due to its mystery, there are a lot of myths around the brain, and I want to disabuse you of some of these myths. For example, it is often said that we use only 10% of our brain. Is that true? Well, no, it's not. We use 100% of our brains. In fact, our brains are constantly in action, and if we are not conscious of it, it's still working. For example, brain scans make it clear that the entire brain is active even during fairly mundane tasks. You may have heard this: some people are right-brained and others are left-brained. This too is a myth. We'll discuss aspects of brain lateralization later on in the class. For example, that the left hemisphere is more involved in language production and comprehension than the right hemisphere. However, we all use both hemispheres, and if you think about it, using only one side of your brain would be an utter waste of valuable neural real estate. What about photographic memory? Do some people have it? Well, it is true that some people have phenomenal memory abilities, and visual imagery plays a significant part in some forms of memory. But we will see that visual information that first enters our brain is nothing like a photograph. So, no, strictly speaking, people don't have photographic memories. Biological psychologists have helped elucidate many aspects of brain and behavior, overturning many of these and other myths about brain function, and enhancing our understanding of brain processes when the brain functions well and when brain processes are disrupted, as in neurological or psychiatric disorders. At least 1 person in 5 around the world currently suffers from neurological and or psychological disorders that vary in severity from complete disability to significant changes in quality of life. The toll of these disorders is enormous in terms of both individual suffering and social costs. The National Advisory Mental Health Council has estimated that direct and indirect costs of behavioral and brain disorders amounts to about $400 billion a year in the United States alone. For example, the cost of treatment for dementia, severely disordered thinking, exceeds the cost of treating cancer and heart disease combined. The World Health Organization estimates that over 15% of all disease burdens in terms of lost productivity is due to mental disorders. The high cost and suffering and expense has compelled researchers to try and understand the mechanisms involved in these disorders and to try and alleviate and even prevent them. Clinical as well as laboratory research are both interested in the applied nature of biological psychology. For example, a clinician may see a patient who presents with schizophrenia, a psychiatric disorder associated with disordered thoughts and hallucinations. In studies of identical twins, with one twin having or showing schizophrenia and the other without the disorder, a clinician may notice structural differences in the brains between the two. Here are brain scans of identical twins, so they share 100% of their genes. On the left is the twin with schizophrenia. And what do you notice? If you look near the center of the scan, the person with schizophrenia has enlarged ventricles, those dark butterfly-shaped areas in the center of the image. Laboratory scientists could then ask specific questions. Did the structural differences arise before the symptoms of schizophrenia, or the other way around? Were the brain differences present at birth, or did they arise during puberty? Does medication that reduces symptoms affect brain structure? By answering these questions, researchers hope to better understand schizophrenia, how it develops, and how it can be prevented and best treated. So in the next couple of slides, I want to show you some areas that are just emerging now in the neurosciences, some of which touches on research that I've conducted myself. So I look forward to some of these discussions. One big area in the neurosciences is the brain's ability to change itself. This is the field of neuroplasticity, which is the physical change in the brain in response to experience or learning. Experience can alter the size of brain regions and interconnections between learning. By engaging in this class, learning the material, you are in fact changing your brain. It's pretty fascinating. The more you learn, the more you retain, the bigger those changes are in your brain. We also know that there are new neurons that are developed in certain regions of your brain through a process called adult neurogenesis. The rate of adult neurogenesis from birth of new neurons in the adult brain can be modified. For example, engaging in cardiovascular activity, going for a jog, can facilitate neurogenesis, can produce more new neurons in your own brain. That's something to keep in mind if you happen to engage in such activities. Behavioral neuroscience spans present and the future. One area of current interest is in what's called social neuroscience. So social neuroscience studies the biological basis of social behavior and the effects of social circumstances on brain activities. For example, examining the levels of testosterone and aggression. One of the founding proponents of social neuroscience is the neuroscientist Matthew Lieberman, who has a book that came out a couple of years ago called Social. I would encourage you to read that book if you want to read a little bit more, and we'll touch on aspects of social neuroscience throughout this class. Evolutionary psychology looks at how natural selection has shaped behavior in humans and other animals. For example, sexual selection of mates and its contributions to brain and development. Another huge field of study that has impacted the discipline is called epigenetics. These are the examination of how genes and environment interact to produce unique characteristics. If you take a look at this sleeping cat, this cat is a calico cat. It gets its different colors because on one of its X chromosomes, this is a female cat, there is a gene expressing hair color that is orange, and on the other X chromosome, that same region of the gene is now expressing black. And due to epigenetic modifications, some of those X's are expressing the orange while others are expressing the black. But we're going to talk in depth about how environment can modify the genome through a process called epigenetics. So gene expression is the turning on or off of specific genes. Factors such as stress, neglect can lead to lasting inactivation of certain genes throughout the lifetime. So this is an area of quite interesting studies, and we'll examine that. Another field is neuroeconomics, which uses brain imaging techniques to identify brain regions that are active during things like decision-making processes. In this realm, there's two known systems: assessment of relative value of choice and evaluation of choices to make conscious decisions. We'll come back to neuroeconomics, but aspects of this touch everything from not only decision-making, but things like drug addiction, mate choice, all of that. Consciousness studies. Consciousness used to be the specific purview of maybe the philosophies, but more and more often individual neuroscientists are starting to study consciousness as an endeavor to understand how consciousness is produced. So consciousness may be described as the state of awareness of one's own existence, emotions, intentions, thoughts, movements, and experiences, metacognition, for example. But in our discussion of consciousness, we'll also be talking about sleep and dreaming, states of coma, persistent vegetative states, and stuff. The neurosciences have a lot to tell us about how we experience consciousness at the level of the brain.